Hello children, this is Austin, your new biology teacher. I hope that you are all safe at home. I couldn't see you now, but I hope that the situations will become normal and I'll see you soon in the classes, right? So before getting into the topic, I just want to discuss something with you. So you would have taken this subject out of your own interest. You would have thought to become a doctor. So I'm damn sure you're going to achieve it. And some would have taken because of your parents' compulsion. Some would have taken because of your friends. So no matter what, if you have interest or if you don't have interest towards biology, I just want two things from you. Okay. So first thing is you have to listen to my class from the first minute till the last minute without any distraction. It may be a PPT, it may be a video, or it may be a class, whatever it may be. Your duty is to listen to it from the first minute till the last minute without any distraction. So this is the first point. Second one is you have to take notes in your science note, and the doubts wherever you get, it should be noted down, and the doubt should be cleared as soon as the class is over. So I hope you understood, right? So these two things, if you are doing it for me, I am sure that you are going to achieve more marks in your exam. So you have already crossed the halfway bridge. You have crossed your 11th standard. Now you are into your 12th. So you have to successfully complete this with more marks, right? So follow these two things. I am very sure that you will be achieving good marks in your exams, right? So now we will get into the topic. So today we are going to start a biology class. We are going to start with a zoology chapter 1, that is reproduction in living organisms. So it is a very easy topic, right? So here first we will see what are the contents that is there in this particular topic. So here we are going to learn about the reproduction process, what it is and why it is important. Then the types of reproduction, because we have different types of organisms, so different organism will reproduce by different methods. So we will learn about the types of reproduction. Then we will learn clearly about what is asexual reproduction and what are the types of asexual reproduction, right? So next, now we will see what is this reproduction process. So we know that reproduction is a biological process by which newer organisms are produced from a pre-existing one, right? This is a common definition that you would have learned. So why this reproduction is happening? What is this? Why it is happening? What is the importance? So that we will see here. So first, new organisms are produced. So only with the help of this reproduction process, we are getting newer organisms in this environment. Without this reproduction, there will be no newer individuals or new organisms that is spreading in this environment. So first point is new organisms. And second one, continuation of species. So particular species will be prevailing in a particular time period, and later the species, if it is continuing to the next time period, it is known as the continuation of species. So if there is no reproduction happening, there will not be any continuation of the species, and the species will get exhausted at the particular time period itself. So we will not get the species in the future generations. So that is what we will tell it as a continuation of species. And next, the variation in species. So we have n variety of species that are prevailing in this environment. So that is what we will tell it as a biodiversity. Have you heard this word biodiversity? So bio means living, diversity means differences. So we have n number of living organisms that is prevailing in this environment. So all this variation is possible because of this reproduction process. And then the exact copies of parent. Exact copies of parent means it is possible in case of asexual method, where you will get the copies of, where you will get the individuals which will be same as that of the parent. There will not be any change in their genetic makeup. Okay, it will be exactly similar to that of the parent. I will tell you why in the next slide, right? And next, genetic variation. So genetic variation, it is common in the sexual reproduction method. So this genetic variation is, there will be some changes in their genetic makeup, and the newer individuals will be genetically dissimilar to that of their parents, right? So I will tell you the reason why in the next slide. And then the survival of species. Survival of species is similar to that of the continuation of species. So we don't have the survival of species unless the reproduction process happens. Okay, so now we'll see the next slide. So the types of reproduction process, as I told you, we are just separating the organisms that are present in the environment into two types. One is lower organisms, higher organisms, that is single celled organisms and multicellular organisms. Unicellular, multicellular, you would have learned this in your lower classes, right? So asexual reproduction is very much common in case of lower organisms, and it is common in multicellular, which is having a simple organization also, right? So basically asexual reproduction occurs in the lower organisms. whereas sexual reproduction occurs in the multicellular organism. It is a higher organisms, right? Now I will just compare these types of reproduction. It will be very easy to learn. If you look at this slide, clearly you can understand what is asexual and what is sexual. See here in biology, a word a comes before a term means it is just opposite to that of. So if you see here sexual, asexual, it will be just opposite to that of the sexual method. When you see the terms like biotic, abiotic, symptomatic, asymptomatic, what is that? See, biotic means living, abiotic means non-living. So it is just an opposite. Symptomatic means showing the symptoms of a particular disease, whereas asymptomatic means they doesn't show the symptoms. So here there is just an opposite meaning, right? So when you see here first asexual reproduction method, it is common in lower organisms and the single parent is involved. It is most important point when you come to the asexual reproduction method. So asexual reproduction method, only one parent is getting involved. There is no involvement of another parent. So here there is no gender like male and female, right? Single parent is involved. So the exact copies of the parent will be produced in the individual. So we will tell that in a term called as clone. Okay, clone means the same. It will be same as that of the parent. Even in the genetic makeup, there will be no difference. Okay, so that is what the exact copies of the parent. So we will tell the term as a uniparental inheritance. So uni means single parent, right? So whatever which is present in that single parent only, it's going to get incorporated into the progeny. Okay, so that is what we will tell it as a uniparental inheritance and no involvement of gametes. So you know what is gametes? Gametes are sex cells. So ultimately only in sexual reproduction you are going to have the sex cells involvement. So here there is no involvement of any, no need of any gametes. So there is no involvement of gametes in asexual method. So now you see the sexual reproduction, it will be very easy for you to understand. So it is common in higher organisms, whereas fusion of male and female happens here. So in case of sexual reproduction only the two parents are involved. Okay, fusion of male and female. So here is two parents are involved and then it results in the genetic variation. Why here genetic variation? I told you no, I'll tell the reason. Why means male, female, they are totally different. They both unite to form this particular individual, newer individual. So here the resulting progeny or the resulting individual will have a combination of both the male as well as the female, both the parents. So there will be some genetic variations in that particular individual. So this is what we will tell it as a genetic variation. There will be variations in their gene, right? Since male is different, female is different, two parents are uniting and they are forming the zygote. So the zygote will have an incorporation of both the male as well as the female. So there will be a possibility of genetic variation in the particular individual. Okay, it will not be exactly similar. For example, you and myself, we are not exactly genetically similar to our parents. So we will have a different genetic makeup when we compare to our parents, right? And next here the involvement of sex cells or gametes. Here the sex cells are involved. Okay, that is what we will tell it as a gamete. So male gamete, female gamete. So male gamete means sperm, female gamete means ovum or ova. So just a fusion of ova and sperm will result in the zygote. So here the zygote will have the genetic variation. So this is what about the types of reproduction, right? Now we will learn what is asexual reproduction alone. As we have learned earlier, the same points, just listen. Offsprings show no genetic variation. So in case of asexual reproduction, there is no genetic variation. It will be same as that of the parent. And the second point, it is common in lower organisms, I told you, right? Like Protista, bacteria, archaea, and even in the multicellular organism which is having very simpler organization. Okay, so these are the organisms that is going to do this asexual reproduction method, and then the exact clones of the parent. So it will be exactly similar. There will not be any dissimilarities between the parent and the individual. So that is what here they are mentioning it as somatic reproduction or somatogenic reproduction or blastogenic reproduction. So what is this somatic and blastogenic? You would have heard about this: somatic cells, sex cells, somatic cells, germ cells. So what is this somatic cells is? It is describing the body cells. So body of the organism, if it is developing, any part of the body or any part of the organism is developing into a new individual, means that we will tell it as a somatogenic reproduction or blastogenic reproduction. Here there is no involvement of reproductive structure. So here it is just the body cells that are going to do the reproduction process. It is just getting divided into two daughter cells. So here we are just telling it as a somatogenic or blastogenic reproduction. I hope you all understood, right? So next, now we are getting into the types of asexual reproduction method. That first we are going to learn about the fission. So what is meant by this fission? You would have heard about this word fission before. Fission, fusion. Fusion means uniting of two things, whereas fission means it is just breaking down. Okay, fission, nuclear fusion, you would have learnt in your physics, right? So here fission means just breaking down or dividing. Okay, it has the meaning of breaking down or dividing. So here if you see here, first, binary fission. So binary means, what is meant by binary? Bi means two. So here binary fission in the sense it is getting, parent is getting divided into two daughter cells. Very simple meaning, right? You would have already learnt in your lower classes. So binary fission, division of parent into two daughter cells. And second one, multiple fission. Now tell me, what is multiple fission? More than two is multiple, right? So here the parent is getting divided into more than two. That is known as the multiple fission. And then the plasmotomy. We will learn what is this plasmotomy in the next slides. So here the parent will have multinucleus, and the multinucleus will get divided, and it will also be incorporated into the daughter as in the form of multinucleate daughter cells, right? So here that is known as the plasmotomy. I will explain you in depth in the next slides. And then strobulation. Strobulation means it is common in Aurelia, where there will be a small fission that is occurring in the transverse region, and the fission will give rise to a new individual. Okay, I will explain you in depth in the next slides. That is known as strobulation. And then sporulation, formation of spores. So this is common in encysted amoeba also, sporulation. So sporulation means the nucleus will divide and it will form spores, and under favorable conditions this spore will be released outside and each spore will develop into a new individual. So this is what known as sporulation method. Okay, so now we will see the first method. So the first method, binary fission, right? So the division of parent into two daughter cells. So first method in binary fission, we are having four types of binary fission. So first one, simple or irregular binary fission. Second one, transverse binary fission. Third, longitudinal binary fission, and fourth one is oblique binary fission. So I will explain you now. First one, simple or irregular binary fission. Take an example of an amoeba, right? So amoeba multiplies or divides itself into two, its nucleus gets divides into two, and then two daughter cells are formed. This is a simple division. But why we will tell it as a irregular means, constitution can occur at any plane of amoeba. There is no particular plane for constriction. So that is what we will tell it as a irregular, because it doesn't have any shape. So constriction may occur at any end. It can occur at the any end. So next, transverse binary fission. So in transverse binary fission, here the breakdown or the fission occurs only in the transverse region. Can you see? This is the transverse fission that is occurring in the paramecium. Take an example of paramecium. If you see here, macronucleus and micronucleus are there. Macronucleus divides by amitosis process, and micronucleus divides by mitosis process. So what is this amitosis and mitosis? Amitosis is a very simple division. Okay, there is no involvement of spindle fibers, whereas mitosis is a involvement of spindle fibers. You have learned the various mitosis stages, right? Prophase, metaphase, telophase, anaphase. So that is little much complicated, whereas this amitosis is just a simple division of a cell. Okay, so macronucleus divides by amitosis process and micronucleus divides by mitosis process. So this is what the transverse fission that is occurring in the paramecium. Okay, next one, longitudinal fission. It is obtained in euglena. So when you see, this is the euglena. So what is an important fact about euglena? It is not an animal, it is not a plant. It is just placed in between animal and plant. Why? Because as a plant cell, euglena has a chloroplast, it can prepare its own food. Like an animal, it doesn't have a cell wall. Okay, so this is what the fact about euglena. So in euglena, if you see, the fission occurs in the in longitudinal fashion. If you see here, a longitudinal furrow appears, and that furrow helps in the separation of individuals. So the longitudinal fashion, this is what the longitudinal fashion that is occurring in the euglena. So the flagella will be initially present at only one daughter cell. Later the basal granules get divided and the newer flagellum grows on the other side of that particular euglena, and just it gets divided in the center in the longitudinal fashion, and now the two euglena will get the flagellum, right. So this is what happens in the euglena. So that is the longitudinal fission. And then oblique fission. Oblique fission, it is not taking in a direct way, it is not taking in a correct way. That is what the oblique fission is. So this is what the word oblique means. So it is seen in Serratium, the fission occurs at any site and there is no equal halves that has been obtained. So here the fission point occurs at the oblique plane. So we will tell this as a oblique fission. Okay. And the second type of fission, that is the multiple fission. So in multiple fission, the parent body will get divided into many daughter cells. So as we have learnt, binary means 2, multiple means more. So here many daughter cells will be formed. First the nucleus will divide itself, repeated division will occur inside the cell itself, more number of daughter nuclei will be formed, and later what happens, see, the cytoplasm will go and will encircle each daughter nuclei. Okay, the cytoplasm will divide and it will encircle each daughter nuclei and it will form a particular structure, and later this cyst will break down and it will release that new daughter cells outside. So inside the cell itself multiple division occurs, division of nuclei occurs, then the division of cytoplasm, particular nuclei will be surrounded by a particular cytoplasm and it will form a particular cell. Okay, like this new cells will be formed inside itself, and all the new daughter cells will be in a form of a cyst. It will be protected inside a covering and later it will be released outside when the conditions are favorable. Okay, so this is what known as the multiple fission. So repeated division occurs inside the cell, more number of daughter nuclei are formed inside the cell, later they are released outside. So this is what known as the multiple fission. You can also learn multiple fission in an encysted amoeba. So why this happens is when amoeba has unfavorable condition, when there is a scarcity of food or when there is a high temperature, okay, when there is a very unsuitable conditions, okay, unfavorable conditions for the particular amoeba to survive, at that time what the amoeba will do is it forms a 3-layered cyst wall around it. Can you see a 3-layer of cyst? So this is a cyst wall. So 3 layers of cyst wall will be formed by this particular amoeba, okay, and this 3-layered cyst will become inactive. Okay, it will be inactive. This amoeba will be inactive. Once the 3-layered cyst is formed, it will be inactive. Now the division of daughter nuclei will be occurring inside that cyst itself, like the multiple fission how you have learnt in the plasmodium. The same way here also amoeba forms a 3-layer cyst. Inside the cyst the daughter cells will divide and later the daughter cells, the nuclei which is divided will get the cytoplasm and it forms a spore like structure. That is what we will tell it as a pseudopodiospores. Okay, so this is the minute amoeba that is present inside the cyst. Later what happens when there is a favorable conditions, when there is a water availability, when there is a food availability, when the conditions become favorable, this particular pseudopodiospores will get released outside and each pseudopodiospore, each young amoeba will get dispersed outside. So this is what known as the multiple fission that is happening in the amoeba. So in case of unfavorable conditions, the amoeba will form a cyst wall, and inside the cyst wall the division will occur and the daughter nuclei will be formed and the spores will be formed. Later this particular minute amoeba will be getting released outside. So this is what the multiple fission in encysted amoeba. Then when you see the process called as strobulation process. So this strobulation process is common in Aurelia. So this strobulation, how it occurs, see the diagram very clearly. So this is what, can you see this formation of small structure in the transverse fashion? Can you see a layers? So this is what the fission that is occurring in the transverse fashion. So in strobulation, several transverse fission occurs simultaneously, giving rise to a number of individuals. When you see this diagram, there will be a formation of effirae budding. So this is what, single effirae. Okay, so this effirae budding happens, and this effirae budding will give rise to fission in the transverse section, and each effirae will be released outside like this. So each effirae will be developing into a new Aurelia. So this is what the actual strobulation process that is happening. Strobulation is nothing but fission that is occurring, and each effirae will get dispersed outside, it will get divided and it will then just form a newer one. So this is what the strobulation process. So here the transverse fissions occurs simultaneously and releases the new individual. So this is what the strobulation. And next one, plasmotomy. So this plasmotomy is very easy. So multinucleated parent, the parent will have multinucleus. Okay, so the multinucleated parent will divide and it will give the daughter cells, and the daughter cells will also have multinucleus. So that is what known as plasmotomy. So the division of multinucleate parent into many multinucleate daughter cells with the division of nucleus. So this is what known as plasmotomy. Okay, so this is what, so the parent will have multinucleus and the resulting daughter cells will also have more than one nucleus. That is what the multinucleate daughter cells, right? So this is what known as the plasmotomy. And then sporulation in amoeba. Okay, sporulation in amoeba, as I have discussed with you earlier, the same point. So amoeba multiplies by sporulation without encystment also. Here what happens is, the nucleus breaks into fragments and the fragments are surrounded by cytoplasm and it will develop a spore case. When you see here, look at the diagram. Nucleus is getting broken down and it releases the small fragments outside. So that fragments are known as chromatin blocks. So these fragments, later what happens, it gets converted into nuclei, and later this nuclei is being surrounded by the nuclear membrane and it forms the spores. Okay, so it is just surrounded by the cytoplasm, it just forms a spores, and later this spore will be released outside and it will be forming a young amoeba. Can you see the diagram? It is very clear, right? So nucleus is getting broken down into chromatin blocks, and now the nuclei will get surrounded by the cytoplasm and it will form the spores inside that amoeba. That spores will be released outside and it will be developing into a young amoeba. So under favorable conditions, okay, so the parent body will get disintegrated and it will release the spores outside. The spores will develop into a young amoeba later. Okay, so this is what known as sporulation in amoeba. The next one, budding process. So budding means what? A small bud will be grown on the parent body. It will be either grown outside the parent body or inside the parent body. So based on this, whether it is present on outside the parent surface or inside the parent body surface, we are just separating into exogenous budding and endogenous budding. Okay, so see here, exogenous means, when you see here, bud is grown on the outer surface of the hydra. When you see here, exogenous budding, hydra reproduces by exogenous budding. You can see a small outgrowth that is being grown here, and this bud will later develop into this entire particular hydra. Okay, so this is what the exogenous budding is. Okay, whereas when you see this Noctiluca, it is internal. The budding occurs inside the particular organism. So here the budding is known as endogenous budding. Okay, so gemmules, when you see in sponges, we will tell that as a gemmules, it is also a internal buds. Okay, this gemmules is also a internal buds. So what this gemmules will actually do is, under unfavorable conditions, what the sponges will do is, it will make this gemmules, and the gemmules will withstand under this adverse conditions. So all these unfavorable conditions, the gemmules will, just like the spore, how the cyst is there, how it is withstanding all the unfavorable conditions, the same way here the gemmules in sponges will withstand its conditions, and later when the conditions become favorable, it will just begin to hatch into a newer individual. So that is what the gemmules. Okay, so when you see about this budding, when in budding you are going to learn about two types. One is exogenous budding, another one is endogenous budding. So exogenous budding means the bud occurs on the outer surface of the parent, whereas endogenous means it is a internal bud. Okay, so gemmules in sponges is also a internal budding. So here these two types you have to be very clear. Okay, so bud is a normal outgrowth that is obtained in a particular individual. So this bud will later develop into a newer organism, right? And next method, fragmentation method. So fragmentation, if you see here, this is a planaria. Okay, this is a planaria. The parent body breaks into fragments, and each fragment will develop into a new individual. So this is the parent body. Okay, it is just breaking down at particular sites here, here and here. So this breakdown is later resulting in a 4 new planaria. Can you see the difference? So fragmentation means the parent itself, this is the parent, so the parent itself breaks its body into fragments. Each fragment will later develop into a newer individual. So this is what known as fragmentation process. So this can be seen in planaria, it can be seen in Spirogyra also. So this is what the actual fragmentation process. So the next one is apolysis in Tania solium. Okay. So what is this apolysis process? You know what is a Tania solium? It is a tapeworm. So it is normally primary host is man, and this Tania solium, what it is doing is it is just shedding its parts. Okay. It is just shedding the gravid proglottis part at the end of the strobila. It just spreads it and it just gives it out. So this process is known as apolysis process. You can see the definition here. The gravid proglottis present at the posterior end of strobila are regularly cut off by the process called apolysis. So like this way it just produces the newer organism. So this is what the apolysis process that is happening in the Tania solium. So the gravid proglottis will be present at the end of the strobila, posterior end of the strobila, you can see this. So they will be cut off regularly. Okay, it will be either single or in a group they will be cut off, and this process we will tell it as a apolysis. So this way it spreads its individuals. So this is what the apolysis that is happening in the Tania solium. And the next process, that is the regeneration process. So with this we will wind up. So regeneration means what? Generating something again, that is the regeneration. So when you come to this regeneration process, there are two types. One is morpholepsis, another one is epimorphosis. Okay, so we have some differences between these two. First one, morpholepsis, the whole body grows from a smaller fragment. Suppose you see this hydra, the smaller, if you cut that particular organism, the whole body will get developed from that smaller fragment. That is what you can see here. The empty part is getting developed into a whole body. So this is what known as morpholepsis. Whereas you see in epimorphosis, if you see, there are two types of epimorphosis. So epimorphosis is normally what means the replacement of lost body parts. If any part is being lost, that part is being replaced means that we will tell it as a epimorphosis. So if you see here, there are two types of epimorphosis. One is reparative epimorphosis, another one is restorative. Reparative means what? Repairing something. Some certain damaged parts alone is being repaired. That is what called as reparative epimorphosis. For this, a good example is human beings. If we have some wound in our hands, so that is what the reparative process. When you have some wound, that wound will get healed within a day or two. So that is what the reparative process of a particular damaged tissue. That particular cell alone will get repaired. That is what known as reparative epimorphosis. Whereas restorative, getting something again, that is restorative. So here damaged body parts can be developed again. For example, the tail of lizard. When the tail of lizard is being cut off, it will again regrow its tail. So that is what known as restorative epimorphosis. So this is what about regeneration process. So this regeneration process is not actually a method of reproduction, because fragmentation is a method of reproduction. Fragmentation, if you see here, I'll just show you again. When you see this, parent organism itself breaks down its own body parts and it gives each fragment into a new individual. Whereas in case of here in regeneration process, only the lost body part is being replaced. So it is not actually an asexual method, but we will normally learn in under the asexual reproduction method. So here regeneration is not actually a method of reproduction. It is just making the lost parts to regrow. That's what. Okay. So in regeneration we are having two types. One is morpholepsis, in which the whole body will be generated from the small fragment. Okay. And whereas epimorphosis means two types. One is reparative, only a particular tissue will be repaired, and that is reparative epimorphosis. Whereas restorative, the last part will be regenerated. That is what the tail of the lizard. Okay. So today in this class we have seen about reproduction process, types of reproduction, in which we have learnt about asexual reproduction and the different types of asexual reproduction. I hope that you have all understood. So all the doubts that you have, please note it down and please make it clear after this class. Okay. So see you soon in the next class. Thank you, children.